Today we're going to be talking about stems, and I want to talk about monocot versus dicot stems. So what I'm going to do is draw a cross section of a stem. We're going to draw in the various structures, label them, and then talk about what those structures do and how they relate to each other. So let's go ahead and get started with a monocot stem. So I'm going to go ahead and draw a large circle just for perspective. And I'm going to go ahead and begin putting in my epidermis cells. And this outermost layer of cells, just like the outermost layer of your cells, is going to provide protection. All organisms on dry land face a huge challenge, and that challenge is dehydration. How do you prevent dehydration? And one of the ways you do that is by lining yourself with a layer of protective cells. So this is going to be the epidermis. And its main job is protection against dehydration and also protection against infections such as germs like bacteria and viruses. We don't want them getting in here. Now, let's go ahead and label this monocot stem. And a big difference between a monocot stem and a dicot stem is going to be the arrangement of the vascular bundles. And a vascular bundle is simply a collection of xylem and phloem. So in a monocot stem, those collections are going to be scattered about the area of the stem. So I'm going to write scattered. So let's go ahead and draw some of those scattered vascular bundles. So I'm going to go ahead and draw a couple here. So there we go. And this is going to be our xylem. These are xylem tubes coming out at you. These xylem tubes are going to be made of special cells called water conducting cells that actually die when they reach maturity. And what's going to be left behind is their bones. And when I'm talking about bones, I'm talking about their cell walls. All right, their cell walls. And those cell walls are going to remain, and what we're going to have is a nice hollow tube for conducting water and minerals. All right, so Xylem Transport of water and minerals that we'll call ions. And the transport is going to be in the upward direction. And that occurs because of the mechanism by which these substances are going to be transported. We'll talk about that in a later video. And again, the cells that make up the xylem, the water conducting cells, are going to be dead. They're going to be dead cells. Once they reach maturity, by design, they die. And they leave their cell walls behind, which serve as the walls of the container. Now, the next thing I want to draw are some phloem tubes. And the phloem tubes, there they are. There tends to be many of them. So draw a couple phloem tubes up here. And they tend to be external to the xylem overall. There they are. And let's go ahead and label those. Those are going to be phloem. And the phloem is responsible for transporting sap, a sugary water solution. So transport sap. And we can transport this sap in any direction. Again, because of the mechanism that's going to be used to transport it, we can transport it in any direction. We want that because sometimes we want to bring the sap up to a fruit way high up in a tree to make it sweeter. Sometimes we want to bring that sap down to the root cells because they're incapable of photosynthesis, but they still need energy. They still carry out cellular respiration, so we need to supply them with a nice energy source. And finally, the cells that make up the phloem, all right, the food-conducting cells of the phloem are going to be live. They're going to be living cells. And that is one of the ways they actually are capable of transporting things, that they are living. So that's going to be useful information later on when we talk about the various mechanisms used to transport things. All right. Each collection here of xylem and phloem is going to be referred to as a vascular bundle. And you can see I have a number of vascular bundles here. Here's 1, 2, 3, 4, 5, 6, and there can be more than that in a given stem, but that's all we had room for here. And these are vascular bundles, and you can see that they are arranged in a scattered arrangement. Now, in between these vascular bundles is not just empty space, but there's cells there. Usually these cells are nice, big plump cells, thin cell walls. I'll draw just a couple here, but they would be throughout the entire area here. And these cells are going to be referred to as parenchymal cells. Let's go ahead and write that down. And their main job is storage and to carry out metabolic functions. So I'm going to go ahead and write storage because that's one of their main jobs. And what things can they store? They can store sugars, they can store fats, they can store water, they can store toxins, they can store pigments. So they're capable of storing all kinds of different things. Now collectively, all these parenchymal cells are referred to as the pith. Now there is no cortex here because we really don't have a layer of parenchymal cells outside here. So there is no cortex here, just pith surrounding the various vascular bundles. Now, that's going to be it for the monocot stem. Next, I want to go ahead and I want to talk about a dicot stem. And it's going to be similar except for a couple things. One of the things is going to be the pattern of the vascular bundles. And another thing, we're going to add in an additional layer of cells called the vascular cambium, which we'll talk about in just a minute. But let me go ahead and erase some of these things. Erase that. And I'm going to write dicot stem. And let's go ahead and erase some of these things in here and redraw them in the proper pattern. So there we go. A nice fresh stem to start with. And I'm going to go ahead and I'm going to start drawing the xylem. So the xylem is going to be these large circles that are coming out at you again. So that's going to be our xylem. And the next thing I want to draw is that layer of new cells I was talking about called the vascular cambium. I'm going to go ahead and I'm going to make them orange. Right? So this is going to be a single layer of cells external to the xylem. And we're going to call that the vascular cambium. And this is embryonic tissue or meristematic tissue. And the function of this meristematic tissue is perpetual growth. These are kind of like your stem cells in a sense. They are capable of undergoing cell division, and when needed, these cells can divide and become water-conducting cells of the xylem, or they could become food-conducting cells of the phloem. So let's go ahead and draw some of those in there. And those cells would be outside here. Those tubes coming out at you. Draw a couple here. And the same thing would go over here. And this is going to be our phloem. So there it is. It's labeled already for us. And collectively, this entire complex, the xylem, the vascular cambium, and the phloem is going to be referred to as a vascular bundle. And you can see that these vascular bundles are arranged in a ring-like pattern. So I'm going to go ahead and write that down here. I'm going to write ring. And again, the vascular cambium, when it divides, sometimes it will produce cells that make up the xylem, and sometimes it will produce cells that make up the phloem. And that way, we're actually going to create lateral growth, or growth in diameter here. And this layer is responsible for making the stem wider and wider and wider. And this is usually seen in dicot plants. Now, let's go ahead and draw some of our parenchymal cells. So here they are, those nice big plump cells with thin cell walls. And they're all over the place here. And they're also in here. And again, job is storage, metabolic functions, things of that nature. And what we're going to do, because we do have a ring of vascular tissue, internal to that ring, we're going to refer to these parenchymal cells collectively as pith. And then externally, out here, we're going to call it cortex. So I guess I'll write cortex right here. And that is going to be it for our Dicot stem.